introduction. I still giggle when people call me Dr. Veatch. I'm getting better at that with time. Okay, so I was asked to tell you a little bit about my story, about how I got to where I am today. So I grew up in Brookline, which is a suburb right next to Boston, where there are lots of professors live that teach at the schools in Boston. So it's pretty, I had a lot of resources, academic resources especially, growing up as a kid. My mom was a doctor, my father was a professor, a scientist, but he died when I was very young, so I didn't get to know the life of a scientist very well when I was growing up, which is probably one reason why I chose to go into it, because I didn't see all the kind of dark side. So I got excited about physics in high school for a lot of the same reasons as the past speaker. I was good at math. I liked being able to not have to remember a lot of things and still deeply understand kind of what I was working on. And actually, I really didn't like biology because my memory for details is just that are not logically connected, completely useless. So I knew that wasn't for me. So I went on to MIT and did my undergraduate degree, also in physics. I should say, so another reason I ended up in physics, or one of the driving factors, is I had this physics teacher in high school who was a woman, who was very sexist and didn't think women should be doing physics. And I didn't understand this at all, but it definitely lit a fire in me to be the best in the class. And to kind of prove that this was something that I was good at, and I was going to show off that I was good at it. And so I kind of blazed through high school with lots of confidence that this was going to be my life choice. So I went to MIT. MIT kind of brought me down a few notches. I don't know, I was still excited about science, and I think I did very well, but I definitely lost a lot of that confidence. And so I left MIT not really sure that I was going to make it as an academic scientist. I also worked on the LIGO project, which was awesome. It got me into the lab. I got to build instruments. I got to travel to the 2 LIGO sites and see kind of how science really works. But I also left knowing I did not want to do that type of science that required this giant collaboration, where I got to do a little piece of a big puzzle. I kind of wanted to have ownership over the whole project. I left out. Oh no, yeah. So I left MIT kind of lacking some confidence. I had applied to grad school anyways, trying to think ahead because I was pretty sure that's what I wanted to do. I spent a year in Portland, Oregon, kind of in the real world, working as an electrical engineer at a company that made lighting control consoles for high schools. So I spent a lot of time programming in assembly, and the first half of it was awesome because I got to learn a new programming languages, turn lights on and off. It was very satisfying. And then the second half, they wanted me to just do that over and over again. And I thought that was the stupidest, most boring thing I could do. And I knew I had to go on and do science. Because if I wanted to keep on learning and be able to justify keep on learning, I had to be in science. Or this is what I thought at the time. There are probably many other ways to do that too. But it was my choice. So I went in the University of Washington in Seattle. My choice of school was based on 2 things. One, it was as far away from Boston as I could get. And 2, I was like following my love interest every day. So not a very well thought out career path, but it was a wonderful, lucky choice for me because I met my thesis advisor, who got me into the science that I'm like amazingly passionate about now, which I'll tell you about. After grad school, I was still hopelessly in love with this person and wanted to stay as close as possible, so I went up to Vancouver, BC, which is driving distance, and I worked in a lab in microbiology. I also wanted to kind of venture into some new area in my postdoc, and I thought this was a good choice. And when I got there, it fell apart for 2 reasons. One, I found myself isolated. I was in a world that the people weren't as excited about the things I was excited about, and so I'd have some result that I thought was super cool, and I'd go tell all of my lab mates, and they didn't really care or understand. And I realized this was not a good scientific environment for me, largely because I had ventured so far off into a different field, and we just spoke different languages. I also got dumped by this love interest, and which is also a wonderful thing, because it opened my eyes that I could go and do what I really should have been doing, which is I did a postdoc at Cornell with a group there that was really doing exactly what I should have been doing. And it was in a lab where I got to learn new biology and immunology, but it was in an environment where they cared about kind of the physical basis of things. And so I was appreciated. I got to make great contributions to the work of the lab, and it was very satisfying. So from there, I applied for lots of funding, and both had some independent money when I was a postdoc, which was great. It gave me some ownership, more ownership over the work I was doing, that when I moved into my faculty position here, I kind of hit the ground running. And so I've been here since 2010, and I'm hopefully submitting my tenure package at the end of the year. That's my life story. It's like a random coincidences that ended up somewhere I'm like so happy with. And I'd like to think that had it not all worked out, I mean, I would have been equally happy about whatever it was I was doing. I kind of see it that way. Okay, a few non-science things about me, because this is a fun opportunity to say it. So in high school, in college and graduate school and my postdoc, I was very involved in rugby. I played, I was a coach. It represented kind of the thing that I got to do outside of the lab that helped me be a real person or a complete person. And I think that's super important. It's been super important for me. Now I don't play rugby anymore. I've become a crazy CrossFitter. I don't know if you know what that is. This is me and my family at our CrossFit gym here. And this is to remind me that I wanted to mention that I'm a mom. So I have 2 kids now. And then I'm also gay. And this is my partner. And actually, at this time of this picture, our second child was still in process, but now he's a little baby. So instead of having rugby kind of to balance my life out, now I have family, which requires lots of balance that I strive to achieve, but I'm not quite there yet. So now I get to talk to you about what I think is so awesome about biophysics. Okay, so I guess I'm the representative of biophysics at this meeting, and I just wanted to be a big advertisement that I think that biology has a ton of questions that are just ready for a physical perspective to come and make huge contributions to. And so my example is one, I'm very interested in membranes, but I think this type of thing applies to many different areas of biology. And I should say, so the biology that I couldn't stand when I was in high school because I had to memorize everything, I've managed to come through the back door. And now, the things I have to remember kind of make logical sense and are connected to me. And so I have a much easier time at kind of learning things. Okay, so I'm interested in membranes. What are membranes? So they're very thin layers of soap molecules, two molecules thick, and they have a lot in common with soap bubbles. So, which we have a lot of real life experience with. So a soap bubble is a thin layer of water that's surrounded by surfactants, detergents, or soaps. And what these small molecules do is they kind of stabilize the interface between water and air. And so they have one side that likes to be near the water and the other side that likes to be near the air. And so that makes this kind of structure last for a while. A membrane is the same thing, but just inverted. It's a small layer of oil, in this case, surrounded by water. And our lipids, again, one side likes to be near water, and then the other side likes to be away from water, so either pointing towards other tails or pointing towards air or oil. And because of that, you end up forming these stable structures. So a little historical sidebar. This field was kind of, early experiments was done by this woman, Agnes Pockels, in the late 1800s. And she was this woman with no formal scientific training, but huge scientific curiosity and the time, I guess, to do experiments using materials and supplies that she could generate from things that you would normally use in a kitchen. And so she did a whole bunch of research, and she wrote it up as papers, and she sent it to journals, and no one took her seriously. And then finally, she sent one of her papers to Rayleigh, who was so impressed that he communicated it to Nature with this excellent prologue. She's considered to be kind of one of the founding people of the field of surface science, and she developed one of the original pieces of equipment that it's still pretty much around today. It was modified slightly over the years, right? And Langmuir got the Nobel Prize for something very similar in 1932. So, vesicles are like soap bubbles, but they're underwater. So think about a water balloon in a bathtub. There's water on the inside, water on the outside, and the boundary of this circle is, or the sphere is a lipid bilayer. They're a lot smaller than soap bubbles, so we use a microscope to look at them. This is me in grad school, actually, taking pictures. And yeah, they glow because we've added a small amount of a fluorescent lipid that lets us see them under this microscope. Okay, so cell membranes, which is really what drives me wanting to understand cell membranes, are really amazing bubbles, okay? So this is a classic movie that I think has inspired many, many people. So this is a macrophage. It's an immune cell, a white blood cell, and its job is to kind of seek out and destroy invaders in our systems. So that's the macrophage. The bacteria is the invader. There's lots of red blood cells the macrophage shouldn't be attacking because that would be a problem. Right? And this movie was taken in the 1950s, and basically it has so much suspense. So the membrane is mediating a lot of the exciting action that's happening on this. Not everything, but a lot of it. Okay, so the membrane is kind of the interface between the cell and the world around. And so it has to sense the fact that this bacteria is nearby. It has to tell the difference between the bacteria and the red blood cells. It has to figure out where it is and go there. Communicate this information to the cell, and then the cell does a bunch of things to help push the membrane closer to where the bacteria is. And now you can't see anything. And finally, when it grabs it, it has to actually bring it in. Okay. So how does it do this? So this membrane is, again, one of these just simple bilayer membranes of lipids. And embedded in it are proteins. And so the proteins and the lipids also work together to do things like detect chemicals that come from the bacteria, push the membrane to deform so that it can follow the bacteria, and then do the grabbing and pulling in when they actually come into contact. Proteins are also cool. Not quite as cool as lipids, in my opinion, but they are kind of bigger and more complicated than lipids. So they have more kind of specificities, more specific functions. So they do lots of great things, certainly. And the membrane proteins tend to be stuck in this two-dimensional plane of the membrane. Okay, so my central question of my lab, and what I've been pushing me for this long, is to try to answer the question, do cells take advantage of the physics of lipids and membranes to actually do some of these amazing things that they do all the time? Hopefully I'll get some hint at maybe that might be true in the rest of this talk. Okay, so back to these vesicles. So there are circumstances where instead of this fluorescent dye being uniformly distributed, it's much more structured. And this is because this membrane can break out into two different liquid phases. So one of these phases is called the liquid disordered phase, one is called the liquid ordered phase. And think of these like oil and water in 3 dimensions. So both liquids, but they don't like to mix. So in my graduate work, what got me so excited, or when I first got very excited about this project, this is what I was working on, is just mapping out the conditions where we see this phase transition in simple vesicles. So these are made of purified components. As a physicist, of course, I ordered them from a company, didn't isolate them or anything like that, and just looked at them under a microscope. And most of the time what happened in the lab was this. I'd make my vesicle of a particular composition, and I would look at it at a high temperature under the microscope, and the fluorescent dye would be completely uniformly distributed. But then as I lowered temperature on my microscope stage, there was a temperature where the vesicle broke out into spots. The spots diffused around. They grew both by collision and coalescence, and by when I lower temperature, maybe they would get bigger. But this looks a lot like a two-dimensional salad dressing bottle or lava lamp or something like that. So I spent a lot of time looking at vesicles like that and mapping out phase diagrams, and I'm not going to talk at all about that today. But the thing that I got most excited about is that there were certain compositions in this phase diagram where it looked very different as you change temperature. So instead of being uniform at a high temperature, we saw lots of fluctuations, lots of flickering. And as temperature got lowered, those fluctuations actually got bigger. The contrast got better. We're lowering temperature more. Now we're very close to where this transition is. This is called the critical point. And now we're in this two-phase region where we clearly have two phases. There's bright and dark, and they're separated by a wiggly boundary. So in this case, it kind of happened all at once. It broke out into spots, and in this case, it was kind of a gradual transition, which is characteristic of being near a critical point. Okay, so this is reminiscent of critical behavior seen in other systems. So this is an example I pulled off of YouTube. So this is CO2 near its liquid-gas transition. And if you start off with a mixture of liquid and gas that's not half and half, and you raise temperature, it's kind of analogous to our vesicles breaking down into spots. You have a meniscus, it just comes up, and it comes back down again when temperature gets back lower. So it's kind of a standard kind of first order simple phase transition. But if you start off with an equal volume fraction of gas and liquid, you see all sorts of other cool stuff. In particular, the mixture gets super cloudy when you go through the transition. And it gets super cloudy because there's pockets of gas and liquid and gas, and they have different index of refraction, so they scatter light. And so this is the equivalent of the flickering that we see in that vesicle. There's fluctuations of one phase and the other. You can also see there's a very wiggly boundary when we go down through the phase transition in this YouTube video. Okay, so the other cool thing about critical systems is that they're universal. So this is CO2, I told you it was CO2. It could be water, it could be something else. It could be a mixture of two different liquids. What matters is that you have to figure out, in this case, the right pressure and the right temperature to pass through this critical point. But once you're there, it doesn't matter what it is all is going to look the same, which is not very typical. So the details of the system don't matter anymore, in some regards. So the example for this in 2 dimensions is the 2-dimensional Ising model. So this is a simple, I'm an experimentalist, I can even simulate this. It's a lattice of spins. The neighboring spins interact with each other, and you throw in thermal energy. And when you do that, you can recapitulate the same types of behaviors that we see in our real membrane system, which is super cool. And not only does it look the same, but you can quantify it and show that it is the same in many regards. So things like the size of fluctuations are the same in these two systems. Okay, so this is really powerful, I think. Membranes I've been talking about so far are simple in that they're made of purified components, but they're still really complicated for a physicist like me to understand, right? They have wiggly tails, there's lots of atoms involved, there's entropy they have to worry about, there's hydrophobic effect, there's ordered water maybe, there's all sorts of stuff going on. And if I had to keep track of each of those molecular interactions to understand what was going on, I'd be hopelessly lost. Okay. So this is really hard. And we do, I think the field is moving forward and has made progress in some of these things, but I don't know. I don't like paying attention to detail. When you're close to a critical point, a lot of things get simple. In particular, so this is the equation that describes the size of fluctuations. It goes as some numbers over the temperature distance from the critical point. TC, and these are things we can measure experimentally. Like 2 things I can measure experimentally instead of a gazillion things I won't be able to wrap my head around. So we can measure the critical temperature. That's the temperature where it goes through this transition. And then C0 is a characteristic length, and then we get that by fitting the slope of a plot. Or we could get it by figuring out what's the size of the molecules that we're working with. And so, because these can be measured experimentally, this makes the prediction of what types of structures we're expecting to see in this system much, much, much more simple than in a system that's far away from one of these critical points. And so this got me very excited. And this is where I was right at the end of grad school. I thought maybe in biology, or so maybe in the simple system we can wrap our head around what's going on, but more importantly, in biology where it gets much, much more complicated, maybe we don't have to worry about all that complication. Maybe we can still use some of these kind of basic organizing principles we've learned to really understand deeply these biological systems. And so that's the dream. And the dream is in the process of being realized, I think, which is exciting. Okay, so does this really happen in cells? So in my postdoc, the postdoc that I really should have been doing from the beginning, they had developed this technique to make vesicles from cells. And so this is a little hard to see, but in this picture there's both cells and vesicles. So the cells are less round and the vesicles are pretty spherical. And what we do is we just grow cells, which is not so hard to do, and then you put some fairly innocuous chemicals on them, but they're not so innocuous that they, I mean, clearly these cells are unhappy. They've decided to shed big blow balloons with their plasma membrane. So they're not so happy, but at least it's not so bad, and it's something we can do easily in the lab. Okay, so here, there's a lot of them. But the big thing is that these are not purified components. This is not 3 components, which we had before. This is all of the components that the cell has decided to put in its plasma membrane, proteins and lipids, and we can study them in the same way we studied our simple systems. Before I got to the lab, they noticed that if you put these guys at low temperature, they phase separate into the same 2 phases we had seen in these model membranes. And when I was there, the most exciting day in the lab ever for me was when this video was collected, which is when I lowered temperature slowly through this phase transition and saw these beautiful fluctuations. So just like in the model membrane, it starts off flickering, now they're bigger than the Planck function. Getting very close to TC here, critical temperature. Now we're below the critical temperature, there's 2 phases, wiggly boundary, and then in this one I actually went back up in temperature, and these fluctuations come back. This is all equilibrium. Okay, so lots of features about these vesicles, just like our model membrane vesicles, we can quantify and show consistency with, or show that they're the same. So if we plot things like the size of fluctuations in the right way on this plot as a function of reduced temperature, the temperature away from the critical point, the data for the model membranes and these biological membranes just lie on top of each other. They also lie right on top of predictions of this simple 2-dimensional Ising model. And for me, this is kind of mind-blowingly amazing that we can use a very simple model to understand these biological membranes. But it's completely expected because this is a universal system. These details that makes these systems different don't matter for things like the size of fluctuations. So this is exactly as it should be. And so this is step 1, I think, for saying that maybe in biological membranes we can use this simple model to make a real prediction. And so here's a prediction that we're making. So we've measured that the size of fluctuations goes as 1 over the temperature distance from the critical point. This is the prediction of this 2-dimensional Ising model. So we've measured these beautiful fluctuations in vesicles within about 1/2 degree of the critical point, and this tends to be around room temperature. So if we were to extrapolate to the growth temperature of these vesicles, we would predict roughly 20 nanometer size fluctuation. So the prediction of this study is that in cells that are doing their normal thing, there should be structure on the scale of about 20 nanometers that looks like these fluctuations. So this is what it would look like. So this is an Ising model simulation, but there's structure in the membrane. This is just 2 components. So the orange circles are a subset of the yellow lipids, and the blue circles are a subset of the blue lipids. And I've shown them both so that you could both see the background and the individual molecules kind of moving around in the background. And so what we're predicting is that there are fluctuations, that there's structure, but it's not particularly, the boundaries of domains, say, are rough. Distribution of domain sizes. The individual molecules diffuse much faster than the background. And that's not because the background's not moving, it's also filled with similar molecules that are moving around, but it just so happens that a yellow spin tends to be replaced by another yellow spin when it moves. So in that sense, these fluctuations can be long-lived. In a real sense, if we time averaged over like a second or so, then this would time average to about a uniform distribution. Okay, so this again was, I think, a pretty exciting observation because kind of parallel to this research in kind of the biophysics world, there's been a lot of excitement in cell biology. about this concept of lipid rafts. The idea that maybe lipids can help organize proteins in cells, and this can help facilitate their function. So this is one of the first cartoons that came out depicting this kind of phenomena, and this is nicely disproven that it's not quite this simple. The current story sounds a lot like the picture that we're trying to tell, the story that we're trying to tell. That in a resting cell, there's some sort of structure, but it's weak, and it's small, and it's short-lived, but that under certain circumstances, it can be stabilized to actually help perform biological functions. And so I'm not going to have time to talk about it now, but our model both predicts this structure, and it gives a couple ways that we could go from here to here. And so I think this is more evidence that maybe we have something to contribute to this biological function. And so my lab does a bunch of things. One of them is to kind of put limits on what we could expect this to do in a biological system, kind of get a sense of what we should be even looking for. And then we go into these systems and actually look for it. And one of the tricks that we use is these new imaging techniques that were recently recognized with the Nobel Prize in Chemistry that are capable of seeing kind of structure, especially in membranes, at about 20 nanometer resolution. And so we're looking to try to take pictures of these structures. Okay, in my last few minutes, I wanted to tell you another story of something that we're doing in the lab that I'm currently very excited about. And that's looking at the molecular mechanism of anesthesia. And I just wanted to point out that all of the data that I'm going to show in this was done by an undergraduate researcher in my lab who is now in med school, but she was in my lab for 4 years. So anesthetics have intrigued membrane biophysicists for a very long time because there's this amazingly strong correlation between the hydrophobicity of an anesthetic and its potency. The very hydrophobic compounds tend to be the best anesthetics, or the best anesthetics tend to be very hydrophobic. And so for a long time, people thought maybe this is just partitioning into the membrane and changing